Hello, thank you so much to Twist having me here today to speak to everyone about some of our research on ways we've been mapping viruses on Earth, ideally someday all of them, or at least all the ones we can sequence, and some implications for Martian missions and also some COVID updates at the same time. So, again, pleasure to be here. Thanks so much for having me. So, I want to start with a bit of background about viruses and microbes, which I think, as most people in this audience are well aware, are everywhere, including mediating plant microbe interactions to, of course, impacting infectious disease. And I think the one that's top of everyone's mind, most recently, of course, in the past more than 15, 16 months, has been, of course, COVID-19 and SARS-CoV-2. And so I want to break the talk into about three sections today. And the first is to kind of do an update of some things that we've been working on in the past year, is to think about all the ways we can bring new molecular methods to bear for better understanding not only this virus but potentially really any virus. So I'll walk you through some of the work we've done on using and developing new tools, some of which has just gotten emergency use authorization over the past year. Actually, now four different tests over the past year, but also some really interesting data we've got from just using RNA-seq in particular to look at the whole transcriptome of patient samples. And so first, I'll start there. Then we'll end up in sewage for a little bit, and then we'll end up on Mars. It should be a little bit of an adventure kind of talk for the day. So I'll start with where we were actually about last March when we started getting COVID samples coming to New York Presbyterian, and we took the first batch of samples we had of over 850 samples and took them and did RT-PCR, of course, because that was being ramped up at the hospital at the time, but also did RNA sequencing to better understand a full transcriptional picture of both host and microbial interactions, as well as a series of controls. And this was just published recently. You can see with Dan Butler et al. In the paper down below. And I want to kind of do a quick snapshot of this paper and how it links to other work that's ongoing today. So one of the first things we did is we looked at, how does this compare to LAMP, which I'll come back to in a little bit, but also wanted to look at environmental samples and also these controls. And one of the first things we did is look at the total RNA sequencing information. And one of my favorite plots from this paper is just looking at the diversity of the transcriptome that we can pick up when we do shotgun RNA sequencing. And this is using a ribosomal depletion kit from NEB, which sees essentially all the human RNA, the bacterial RNA here in green. And of course, for those that were RT-PCR positive, we can see these orange, kind of high, medium, and then low amounts of the virus present in these patients. Sometimes very trace amounts of the virus is present. And what's extraordinary is we could see in some cases that looked like they were maybe PCR negative, they actually were positive, but very rare would we see false negatives. But in some cases, we would see as much as almost 80% of the actual sample was actually SARS-CoV-2 RNA. And so, this is intriguing because we had these distinct viral profiles, but we then wanted to see, well, how often do we have other infections that are often present? And so here we can see it's relatively rare. If you look in this plot here, this shows patients with no SARS-CoV-2 versus low, medium, or high, as measured by RT-PCR NGS. And also on the very right, this kind of pink subsection are those that actually had other respiratory pathogens you can see picked up here. But in general, the presence of these vertical lines showing positive for SARS-CoV-2 and other viruses was relatively rare. Most common at the time was with influenza A, although what's an odd benefit of everyone wearing masks in the past year and social distancing is there's very little influenza A left to find in these days, actually. But we could see, and this was buttressed by other publications, that co-infection is relatively rare, but this does give us the opportunity to pick up other pathogens when you just do total RNA sequencing. And because, again, it's a total RNA-seq, as you remember from just this plot I just showed you, we can see bacterial RNA as well. We could use this to do basically a metatranscriptome profile. And then we looked at the differences, again, between high, medium, and low patients for SARS-CoV-2, as well as those with really distinct other pathogens. And we can see here in red are those species that were statistically and significantly different between those other categories that didn't have the infection. And you can see this was Klebsiella, some Staph aureus. There's kind of a microbiome disruption that we could see as a consequence, or at least associated with this infection. So it raises interesting questions about the sort of order. Does this microbiome disruption lead to a greater chance of getting infected, or did the infection directly create this perturbation? We don't know that yet, but are looking at this currently. And you can see here, this assay lets you actually examine all those questions because you're looking at the RNA of viruses and bacteria. We also did phylogenetics of the virus. And since we have RNA from the humans as well, we then got to look at differentially expressed genes that were in the humans. So in particular, we looked at those genes that were upregulated, you can see here in orange, or downregulated, you can see here in purple, and looked at the degree of their change on the x-axis, or the statistical significance on the y-axis. And we could see there were a lot of these genes that were in pathways that would make some sense. So if you see viral response, you see interferon-stimulated genes, We can also see a lot of genes related to the host response to an infection. And if you did them by pathways, again, some familiar networks like antiviral modules, gamma response, interferon gamma response. But the really surprising ones were olfactory signaling or hemoglobin complex. These really intriguing implications of how these were disrupted pathways, not just present, in response to normal viral infection, but also leading to some of the loss of sense of smell and taste. And so you could actually see that in the nasopharyngeal swab data. And so we did build a website where you can actually go in and query specific genes and make some of these same plots and heat maps and pathways here at covidgenes.wild.cornell.edu, also featured in the paper. So this was, looking a lot at the RNA across different species, but we also wanted to use some new technology to look at spatial transcriptomic or spatial omics methods. In this case, this was a really great collaboration with Rob Schwartz and Alan Borzek, who's the pathologist who did a lot of the autopsies, to get basically 39 patients' autopsies completed where we did geomic spatial profiling of these samples as well. And here we had oligoconjugated antibodies where you'd scan a region of interest, cleave off the antibodies with essentially the oligos, and then drop them into plates and sequence them to reconstruct spatially what was present in these different slides. And here you can actually see kind of this rim of a canyon. You can see the ACE2 receptor and some subtle presence of TMPRSS2, also DNA here stained in blue for DAPI staining. And when you zoom in, you can see like clearly there's disruption happening in some of the alveoli and other areas of the lungs. And we can see not only what was happening for the localization of different markers, but also the proportions of cell types present that we could deconvolute also showed quite dramatic differences. In particular, we could see for COVID patients, you could see an enrichment of neutrophils, or you can see here this dark green macrophages really pronounced, this really strong infiltration of these, sort of new, immune cells that normally might be transient or really persistent and created a lot of continual inflammation in these patients. So really disrupting and eventually leading, to destruction of many of the tissues and even likely contributed to their unfortunate demise. So in this case, and this has now been replicated by other papers in particular, but we also could see from the autopsy samples, we could pick up trace amounts of the virus in other parts of the body, but it was very low levels, much less than what we'd see on, say, nasopharyngeal swabs. But in all cases, we could see this enrichment of the subgenomic region of the virus. You can see here 3', this sort of stronger coverage. You can see this, the normalized coverage in each one of these tissues that we could pick up there. And so this begs the question of it'd be great if we had better methods to pick up, the virus from these tissues where it's hard to pick up, like in tissues, or I'll come back to this in a little bit, like sewage. And so we're very excited to look at capture and amplicon methods. And in particular, we just recently got emergency use authorization from the FDA, working with Twist and Conflict of Interest Note. I'm a co-founder of Biotia, but very proud of the work we just finished of actually starting to get, our research use only product out onto the market. And then within a few months, actually getting it through the FDA and now launched out. So anyone can use this product. And this is, again, a Twist hybrid capture assay. So basically what this is, you collect your specimen, convert it to RNA, RNA to cDNA, prepare it for sequencing to get it in TrueSeq compatible libraries, do the enrichment. And then we've been doing a lot of our sequencing on the NextSeq 550, and we have a pipeline that gives you the output of your variants as well as your positive negative calls using our algorithm I'll describe to you shortly. But basically could give us, this COVID DX pipeline gives you a full readout of what's present in your sample. And in particular, it's actually something that's fairly manageable to do. There's 5 different modules that actually involve sort of nucleic acid prep, cDNA synthesis, the library prep, the capture, and then sequencing. And so you can see here, this is the sort of full workflow, but it includes, when you buy this workflow, from Twist, also the Biotia pipeline is actually embedded inside of it. So you get this all, sort of free of charge, if you will, once you buy the entire packet. You can basically run this, take your FASTQ files, and then upload it onto our portal, which is it also went through the EUA. So the FDA did a full sort of cybersecurity audit and sort of workflow analysis of this EUA product. So you can actually get an end-to-end, a full stack, basically informatics package. It takes the raw sequence files and gives you essentially these really nice reports. So you can see there's a preprint that's in review about this protocol. This is again from the COVID DX software. And we got about 97% PPA and 100% negative percent agreement, down to just below 1 copy per microliter. And again, you can see here, what do we see for variants in your sample if you upload one, and what do we see for what's the frequency of these variants in GIS-Aid. And we wanted to actually look at this, not just in the context of, okay, can we use this to pick up variants, which is not surprising, but what else could we do? We could say, what's the depth of coverage, even when you start to get lower amounts of the virus, and as a function of CT value, what do we see? And we could see, at least 5x coverage across almost all the genome, And for most samples, once you get to a CT value of 30, 25, starting to be below that range, getting really 99% coverage across the whole genome, and you can really anything below 30 give really good coverage, which is not too surprising for those who've been in sequencing for a while. But so that was intriguing and not too surprising because more virus present, you'll obviously get better capture of the virus. There's more molecules for you to capture. But I think the interesting thing from this paper is that we also picked up unique variants that we didn't see in JS8 that were then validated with Sanger sequencing. And so these are basically distinct mutations that we can pick up because the probes are long. Essentially, they're 120 bases and they have a lot of tolerance for mismatches, so they can pick up other mutations that might not be picked up on other assays. And so, we could pick up, but we can still see the common variants, and this includes ones, for example, that are now the predominant strain, for example, in the US is 117. You can see we can pick it up here with some synthetic controls, and we've also seen it in patient samples. You can see here, given these known mutations, as well as this one that's the deletion we can pick up in 117, can readily be picked up with the hybrid capture protocol. And so it's very exciting, very proud of this work with Biotia and Twist, and we also now have the software has been updated. So you can actually see phylogenetically where does your sample land, and then where does it actually show up again, the variants relative to everything that's known for known variants as well as their clades. And in particular, this is the variants are for research use only, but there's ongoing discussions at the FDA about how do we actually get these variants out to be used more routinely and have a validation pipeline for them. And so I think even though all tests actually still to date are really qualitative yes/no tests on the market, ours is one of the very few NGS tests that has built into it an entire protocol to give you the variants. And also, again, our entire Biotia pipeline here, the COVID DX, was built with a full cybersecurity audit from the FDA that is now part of the EUA. So really excited to have this out on the market for people doing further testing. Okay, so that was a little bit of work we've done on some assays. There's also a LAMP, which is something we've been playing with a lot in the past year, which if you're not familiar with it is basically a loop-mediated isothermal amplification. So instead of just having 2 primers that target one region, this has 6 primers usually that targets either DNA or RNA, and you can add in a warm start reverse transcriptase plus a strand displacing DNA polymerase. So it enables you to actually quickly go from a target RNA to cDNA to double-stranded DNA, and you can put it into reaction. And we first started playing with this just over a year ago, where we could actually see the reaction starts all pink. This is with a Twist Bioscience actually RNA, and also we can see here with this controls, you can see when you start to add in more time and just watch what happens, the colorimetric shift occurs, and we can see the LAMP reaction was complete then at 30 minutes. And so it's actually, if the virus is present, it switches down to a yellow color, whereas if it's pink, the phenol red stays red. And what's exciting about this is these Twist synthetic RNAs were used for the beginning and still today continual validation of these LAMP protocols, where we can actually see some clinical samples as well as Twist serial diluted titrated samples. We could clearly pick up the presence of the virus down to 100 or 1,000 copies of the virus. And this led to work that we started giving this protocol to anyone and everyone that we could. This led to an EUA with Color back last year for doing LAMP in their high throughput lab. It's also a full protocol that's now from NEB. And I serve also as an advisor to NEB, so conflict of interest note there. But I'm very excited because it was something that was a beta protocol and just got it going up onto the market very quickly. Again, leveraging sort of the Twist controls to make sure we were getting this right. It's also now a global group, which is an open group of people sharing methods, protocols for deploying LAMP at community centers, high throughput testing labs, variations of LAMP now for TB and other targets. And so we also then deployed this actually in the past year in my hometown in Wisconsin. They were running out of protocols and had no methods for testing at the time. So we actually set it up so that the firefighters and police force could start to run some of their own protocols. You can read about this here when working on saliva in a former boxing gym, which you can see here looks a little bit like a Breaking Bad episode, but actually it's worked fine for a clinical lab, for a pop-up lab. And we also got it working at Weill Cornell in the emergency room, so testing patients right when they would come in to essentially to get tested for COVID and also to further clinical workup. So this has been, a really intense and crazy year, but really, productive in terms of EUAs and work that we've done. But I want to take the last, or sort of the middle part of the talk to say, well, what if we'd been looking all along? What else could we have been finding? In particular, there's a network of researchers called Metasub, which is the metagenomics of subways and urban biomes, that historically has been looking for microbes just in urban environments. But we realized about a year ago, we started actually sampling in hospitals, sewage, and public surfaces because we had a network of people that could begin to share data. And so globally, we launched a project to actually start this in 2020 and have so far actually sequenced shotgun RNA sequencing from actually about you can see 2,200 samples, and so this is an open consortium. So if people want to play with this data, they're welcome to it. And we so far have seen actually that you can actually see different cities at different stages in their pandemic would have varying amounts of the virus. In some cases, when it was peaking, we could see more of the virus actually present either in their hospitals or in some cases in their city centers. And it wasn't just SARS-CoV-2. We could also pick up, for example, other viruses. We could see this fragmented but informative RNA. This is the coverage statistic we could see picked up. In this case, this is from a hospital samples on the right and some from sewage as well here on the bottom. And we could pick up other viruses like influenza. We could see different phages, other RNA viruses. Really, this shotgun data gives you a broad snapshot of really all viruses that are present. And so it begs this question of like, if we had been looking all along, maybe we could have seen the viruses before it got too late. And so this led to a recently funded project with the CDC and with the NIH to actually build, Miami and New York City and Wisconsin wastewater sequencing, and also in partnership with the Metasub Consortium you can see here. And so there's a collaborative work between myself and Stefan Schurer, Helena Solo-Gabriel, and George Grills. And so really a great collaboration to start to embed this into cities. And so what we've been piloting out essentially these dashboards are from Miami-Dade and also from New York City, is to basically say, let's take a look at new cases, hospitalizations, and deaths and see what are the cases per county and build out some of these dashboards that can start to link the public health data with what we see for the molecular data. And in particular, what we've seen so far is been testing on campus, sometimes daily and hourly and weekly sampling across the Gables academic campus. You can see here, this is led by Dr. Solo-Gabriel. And also comparing this with ongoing testing of the students, also positives by dorm and by dorm room, and linking this to an IRB that we have active that also collects the medical records and matching that to county level data. And so what we've already seen that's really extraordinary, I think, is that the sewage data is there's a really close correlation between the positive cases in the county, but also in some cases, the sewage data here in blue can actually precede the positivity in the community. So it actually means that this really could be an early warning system if done right and done continuously. This is from our own data, and so we've been ramping this up more for increased sampling. In particular, we were doing weekly sampling since last summer. We've now been ramping this up to start doing some daily samplings, and we'll have a few of the targeted sites that do hourly sampling. And what we're doing with these samples is kind of a collection of everything I've just told you about for the first part of the talk. So in particular, we're going to be taking all the samples and doing a sort of a multi-omics profiling for the wastewater. In particular, doing variations of LAMP, RT-PCR, doing shotgun RNA sequencing, and also doing some deep targeted and hybrid capture sequencing. So a mixture of basically everything I've just told you about, and also then to confirm strain identification across all these labs. And so I think what's really exciting is we can see these data, really starting to inform and link to public health goals. And the other thing that's exciting, I think, is we have a management system that we've built actually to keep track of all this data. So in particular, if you look at this website here, you can go to Pangea and look at all the sewage data that's not becoming public. So it lets you search by sequence information, also species, everything that we found. And so we built Pangea originally because we wanted a real great system for many collaborations in our lab, but also because we wanted a hub for the Metasub network. And so our goal here for this network is to really build out these global metagenomic maps and also to track antimicrobial resistance, and also to use this information to scan for basically new molecules, new peptides, potentially new drugs. And over the past 5 years, we've actually had a really continual collection process. And so it's been, these are many of the sites across Metasub that over the past 5 years have continued collections for DNA and RNA, still really hunt for new viruses. And again, it's in, really high touch urban environments, but these are places where we find all kinds of things. And I'll show you shortly. This is really the beginning, I think, of what you could flag as a intriguing snapshot of like a planetary genomics. We're actually scanning and monitoring and sequencing and mining the data from around the world at a planetary scale, or at least across over 100 different cities. And this was really inspired by a study that we first did back in 2013 and 14, where we did the first snapshot of New York City, when we swabbed across the city, annotated, sequenced it, brought them all back to the lab, extracted them and sequenced them, and then trimmed these all. And actually, when we did all the alignments to any known species, we found that half of the sequences matched no known organism. We didn't know what they were. A lot of them were bacteria and viruses, but many of them were completely unknown. And so this really meant that half the world under our fingertips was unknown, and actually still for the most part is today. And so I've been pleasantly haunted by this figure from this paper, is this unknown organisms that are really literally under our fingertips. And it kind of begs this question of who is there and what are they making? Can we learn from these creatures to figure out essentially, do they have any impact threats for public health? Are they just part of the normal microbiome? Can we learn from them about their adaptations to environments that are cleaned or sort of stainless steel surfaces? And does this have relevance for other projects? For example, things that could survive harsher missions, for example, going to Mars. I'll come back to that. And so, with funding from the NIH and also from the Gates Foundation, is to actually develop out this global map. I'm very excited that actually coming out tonight and also be online tomorrow is a paper that we just, here's the preprint for the paper, but basically is a global genetic map of the urban metagenomes and antimicrobial resistance. And this data has led to a threefold increase in the size of the tree of life, been used by our group as well as by others. And this includes actually 11,000 new DNA viruses, mostly phages, that we've just found and released with this paper. And so what's really exciting about this actually is it's not just finding new viruses, but also we found, about 4.3 million novel peptides and over 800,000 new CRISPR arrays. This is work with Jonathan Gutenberg and Omar Abdullah, and also with Arbor Biotech. Is basically with at least even 1.5 kb contigs, we're finding all these different fragments that are not matching anything that's been known before. You can see all the novel contigs from de novo assemblies. What's the sort of agreement between it? Is it 5% or 20% divergent from any known peptide? And you can see Tokyo has the most novelty for whatever reason. Actually, you can see here we think this is maybe because of its cosmopolitan nature, but it is also a very unique city that on average, half of the new peptides are at least 20% divergent from any other known peptide. So a lot of novelty there, whereas New York City actually is only 8.4% are really, I think, at least 20% divergent of their peptides. So not as much novelty there. And so what's intriguing about this is, if you sequence just even the world in our cities, you can, and we have found novel microbes, novel species, novel peptides. And what's interesting is using these exact same methods of, sequencing de novo assembly and sequence comparison, we've also found them in space. Like we just published a couple months ago, essentially new bacteria that have evolved enough on the space station to be classified as a distinct species. And the reason I bring all this up is because, we're finding all these new potential viruses, all these new subpeptides, even finding new ones on the space station. But I describe this as a quest to find all the world's viruses because I want to see what happens, for example, when we're making a robot that would go to another planet. Could we actually, would we know a new virus if we found it, if it was on Mars, for example? And while this seems like a hypothetical question maybe only a few years ago, it is actually something we'll be able to address in a matter of a few years. Because you can see here, this is the Jet Propulsion Laboratory clean room where they actually construct the robots that go. This is the spacecraft assembly facility of the SAF. And here in particular, this is the Perseverance rover as it was being built, and you can see engineers get excited too, like they're people like we are. Engineers are people too. They would take a picture with the robot that they're going to send to Mars. I would do the same thing, I think, if I was about to send something to another planet. What's exciting about this, though, is that this fun picture here is the thing about it, there will be samples that the Perseverance is going to go collect eventually. It's going to bring samples eventually for a return mission. So the goal is to kind of tool around Mars. These are pictures from Mars here. Look around the environment for a good place to start do some digging. Again, a picture from Mars. And also right under the robot from Mars, it'll find a good spot to dig into the soil. And this is the regolith here. And eventually capture up some samples, put them in tubes, get them ready for a sample return mission, and stare back longingly at Earth, waiting for them to be picked up. And this is a picture of what it looks like on Mars looking back at Earth. And so eventually, hopefully by the year 2028, they'll be picked up. And then by the year 2032, the Mars sample return mission should, in theory, bring them back. And so one of the very first things we'll do is, of course, characterize these rocks. These samples will be preserved in a location to make sure they don't potentially infect anyone, what's called planetary protection. But we'll also try and sequence them and characterize them and look for potentially, peculiar kinds of life. And, but then one of the first questions will be, if we see a virus, is it really a Martian virus, or is it just something from like the microbiome of the skin or the bodies or the clean room of some of these people? And so to begin to address that question, we've combined some of the work of Metasub and sequencing and assembly with also a deep characterization of the clean room for JPL and built what we call as a comprehensive metagenomics framework for planetary protection. So we could see what are the species that look like they might be novel that can survive in the clean room, which you can see here we've done with de novo assemblies, and what characteristics do they have? So in particular, do they have genes for DNA repair or motility or spore formation that might be relevant for planetary protection? So we wanted to look at the ISO clean rooms, and if you're not familiar with the ISO clean room standards, they go from 1 till 9 generally, and these are essentially the standards that are used for a lot of clean rooms, where it means what is the maximum size of the different particles you can have per per cubic meter, and you can see here the different sizes of them. Obviously, if it's a lower clean room, it's very very strict, and less particles are allowed of continually smaller size. And so we characterize the clean rooms of JPL, which are between ISO 5 and 7, some of the different variations of the clean rooms, to see what's there and compare them to negative controls that are in these clean rooms to see what organisms we could pick up. And so from this paper we just published, this is a number of reads assigned to each taxa. You can see some of the normal flora that you'd normally expect to pick up in the clean room we could pick up here. And you can see these are different categories of the samples and different ISO rankings of ISO 5 or 6 to 8.5. And you can see when you get to the higher ISO ratings, of course, there's more particles that are allowed through, so you'll see more species and then more abundance of them. And if you look at the characterization of these species, though, and you kind of put them together, what's interesting about this is not just you'll find more reads, these were essentially prepped the same way, sequenced to the same capacity, and you'll see, more organisms present, but also they're different in terms of their type as well. So if you go here, actually, let me go to this one first. If you look at the ISO organisms first in the higher ISO, which have more species, more reads, you can see a lot of them are ones, though, that could potentially be psychrophilic or ones that could be radiophilic even, or classified as extremophiles, things that could potentially survive, things like Methylbacterium radietolerans, things that could make their way through the clean room, or at least we're picking them up in the clean room. And that means if they manage to make it anywhere on the spacecraft, although unlikely because the cleaning protocols are really rigid, but if any of them did, these would be the candidates that potentially could be flagged as ones that we'd look for as maybe false positives or ones that could maybe even make the trip. And again, this entire pipeline is online. And so if you then go to the ISO 5 species, there were not as many of them, but still some of them classified as things that could possibly resist sort of the cleaning protocols and maybe even make the trip there. And so when we look ahead, essentially what we've built from this is essentially a map of everything that's in the clean room, everything that's in, let me go back to this, everything that's in that could survive the trip and even on the way back, maybe survive the trip all the way back to Earth. And this basically has given us a global Kmer index as part of the project in Metasub. This is built by Andre Colles and Gunnar Reich in Metasub and also in Switzerland. That makes it such that you can type in the sequence of anything present ever observed on Earth and find where it is and where it might have ever come from. And we can basically, this index is what we'll use when something comes back from Mars. We'll quickly be able to tell, was it seen in the clean room? Was it seen anywhere else? Right? So as long as it's a nucleic acid that we can sequence, we can quickly and readily annotate those species, characterize them, and to discern, was this something that is from that clean room or not, or anywhere else in the world, and gives us really a truly planetary scale genomic map. And so I think this kind of work, which is really just the beginning of some work we wanted to do in the lab. If you go here, actually, I'll show you a little bit of how it looks. You can see here my calendars. So it's basically everything that's ever been sequenced, you can see across the index, and you can search essentially, given a certain cutoff. You want to say, I want to look for, I've seen this sequence, I'd like to know, has this been seen anywhere on Earth before? And if so, where has it been seen? So this shows you the ability to link to the metadata, in particular from Metasub, as well as from other sites in any other database. And also then you can do alignments of them to see how do they actually look, and also visualize them if you want to play with the De Bruin graph. This also gives us an ability to do so. It's kind of a fun interface here to play with the De Bruin graphs as well. And so when you think about what this is, again, this is a planetary scale genomic map of everything that's there. And the reason we want to continue to do the sequencing and the assembly and the annotation and the mapping of microbes is because at the microbial level, this is something that's really just a mirror of what's happening at the macroscopic level. In particular, we know that, for example, in the past 50 years, 1 out of every 4 birds are dead that used to be in North America, or at least we can't find them anymore. And this is not the first time that species have gone missing or extinct. In fact, there's been probably 5 mass extinctions in the history of the Earth. Over the past 500 million years, the world has sometimes had polarized caps, sometimes it hasn't. But every time we have a dramatic change in temperature, we've had a massive loss in number of species. Anywhere from 75% to 96% of all species have been lost. And then we're worried about a 6th extinction potentially happening in today's era. And if you look and you kind of zoom out from this 500 million years, but if you zoom out and take this in the context of the history of the Earth and the likely future of the Earth, we're really in this sort of nice but temporary temperature canyon. We've got liquid water on the planet. It's not so bad. But eventually the sun will probably get hot enough to boil the oceans. And so we do have a bit of a risk, not just for life here, but eventually life in the future. And learning the genetic adaptation lessons of every organism on Earth with these sort of planetary maps and even eventually interplanetary maps might help us understand how life can adapt. And more importantly, I think it helps us actually serve as guardians of life. And particularly if you think of, what did you think of a food chain when you were a kid? You learn that there's 3 kinds of organisms: producers, consumers, and decomposers. And that's really it. But I think there's a 4th kind of species that is uniquely represented by humanity, and that's just that the guardians, people who are aware that there is extinction as a possibility, and only we can actually, I think, do something about it. But of course, only humans are extinction aware. Only humans sometimes have created extinction as well, but we're the only ones that can actually, I think, prevent it as well. It gives us kind of a newfound duty and purpose, particular not just sequencing, not just mapping, but using this also as a functional genetics conservation effort and really understanding the evolutionary toolbox that's inscribed in the DNA of every species that we sequence, whether it's microbial or macroscopic. All of them are really have fruit that we can use to better understand life here and how it might be able to survive elsewhere. And so, and then as a quick plug, since I just published this, is I make the same argument in the book that just came out. So I think this is a preserving and mapping of genetics is not just a exercise in fun or curiosity, although it is. It is also, I think, an innate manifestation of our duty to map and preserve species on this planet and someday potentially others. And so with that, I want to thank very much Twist for having me here today. Thank collaborators, colleagues, and friends of many, many places, too numerous to list, but this is a short list of some of them. And of course, I want to say the biggest thanks is to the patients, to their families, and with the sewage sequencing to what they may leave behind. And I'm happy to take any questions. Thank you so much for your time. It's a pleasure to be here.